All right, let's dive into a really cool and incredibly powerful idea from a recently reconstructed manuscript. It's all about finding a smarter, faster, and frankly, more beautiful way to measure the very building blocks of our world, molecules.
It all starts with a question, just like this one. I mean, whether we're talking about medicine or creating the next breakthrough material, progress pretty much always depends on how well we can understand and predict what molecules are going to do. And that boils down to something surprisingly fundamental.
Yep, turns out it all comes down to math. A puzzle, really. To get a handle on a molecule, you first have to be able to measure it with incredible accuracy. And for decades, the way we've done that has been, well, let's just say it's been a bit of a brute force, messy affair.
So what is the actual problem here? Why is just measuring a molecule so difficult in the first place? Let's break that down.
Okay, so here's the core challenge. A molecule isn't just a single dot, right? It's a complex, three-dimensional cloud of electrons. And to figure out its properties, like its energy, you basically have to add up the influence of every single point in and around that cloud. That whole adding up process is a mathematical thing called an integral, and doing it over an entire 3D space is computationally very, very expensive.
This slide really gets to the heart of the matter. The old way, the hard way, involved this thing called partitioning space. You'd kind of slice and dice the molecule's volume into these fuzzy, overlapping zones for each atom. It works, but man, is it complicated. The Hall and Rees idea, a smarter way, is just radically simpler. Forget all the carving. Just use one single unified grid of points for the whole shebang. And that brings us right to the core of this innovation. What on earth does this new kind of grid look like, and what really makes it so different?
This one sentence from their original paper, this is the revolution. No space partitioning is required. Just think about that. Forget all the messy slicing and dicing. The whole idea is to just sidestep that problem entirely. So how do they pull that off? The idea is so elegant, it's almost shocking. It's just two steps. First, for every single atom, you generate this perfect, highly optimized little cluster of measurement points, or nodes. And then, step two, you just combine them. You take the grid for your carbon atom, the grids for your hydrogen atoms, and you snap them together into one big grid for the whole molecule. That's it. Seriously, that's the idea?
Okay, let's make this real for a second. This here is the simplest version of their atomic grid, a 7-point grid. Look how clean and symmetric that is. You've got 1 point right in the middle at the nucleus, and then 6 more placed perfectly along the X, Y, and Z axes. It's just a beautiful, simple, and powerful structure. But okay, what's the magic trick that lets you combine these and have it all work out?
Well, here's the secret sauce. It's not just where you place the points. It's about a special set of mathematical functions that are custom built to go with this grid. They called them hybrid functions. So the key to everything is this thing called the exact interpolation property. Now this sounds technical, but stick with me because this is the whole magic trick. For every single point in that grid, they create a special function that has this amazing quality. It's perfectly on, like at 100% power at its own grid point, but it's exactly off, a value of 0, at every single other point in the grid.
The absolute best way to picture this is with an analogy. Imagine each measurement point is an actor standing on a dark stage. These hybrid functions are like a set of perfectly engineered spotlights. Each spotlight is so precise that it illuminates only its one target actor, and it doesn't spill a single drop of light on anyone else. Everyone else stays in total darkness. That perfect targeting is what makes the whole method work so cleanly.
Of course, a clever idea is one thing. A cool analogy is even better. But the big question is always, does it actually work when the rubber meets the road? Well, Hall and Rees put their method through its paces. Let's see how it held up.
And they didn't just mess around with some abstract models. Nope, they went right for the classics, molecules that every chemist knows and works with. From the simplest possible molecule, hydrogen, to the incredibly important water molecule, all the way up to the famous ring structure of benzene.
And when you put it side by side with the traditional methods, the difference is, well, it's stark. Those older grids often need 50, 100, even 200 points per atom to get the job done. The Hall and Rees method? It's achieving incredible accuracy with as few as 7 or 15 points. Plus, it comes with a bonus the others don't, a mathematical guarantee that it's perfectly exact for a whole class of functions.
So what does using way fewer points actually get you in the real world? One word: speed. A lot of speed.
For a molecule like benzene, which is a benchmark for these kinds of calculations, their method was up to 20 times faster. Now, that's not just a tiny improvement. That's a potential game changer. A speed-up like that means you can tackle problems that were once just too big to even try.
So the verdict from the data is crystal clear. This new approach isn't just some neat theoretical idea. It delivers what's called chemical accuracy, which is the gold standard you need for results to be useful. But it does it with a fraction of the computational effort.
Okay, so we've got a faster, more elegant, and more robust way to measure molecules. Why is that a big deal for those of us who aren't computational chemists? What's the bigger picture?
So let's just recap the big wins here. It's mathematically guaranteed to be exact for its basis set. It uses a bare minimum number of points, making it super efficient. And it completely avoids that whole complicated, messy business of partitioning space. It's just a fundamentally smarter way to do it.
And here is the big so what. You have to think of these calculations as the engine that drives modern science, especially in chemistry and biology. If you make that engine dramatically more powerful and efficient, you're not just improving one little part, you're basically accelerating the entire process of discovery itself.
What does that actually look like? Well, it means being able to screen thousands of potential new drug candidates on a computer instead of just hundreds. It means designing brand new materials like better batteries or stronger alloys with properties you've predicted before you even step into a lab. It just lets us understand the world around us on a much deeper level.
Ultimately, everything is molecular. Our bodies, our planet, our technology. The Hall and Rees method is essentially a sharper, faster, better ruler for measuring that world. And that leaves us with this one really exciting question. With a fundamentally better tool in our hands, what new things are we going to be able to build?